notice that fine, dusty white film on the leaves of your garden plants? Or maybe on a wild berry bush? It looks almost like someone spilled flour, right? It's incredibly common.
Mm.
But what if I told you that unassuming white coating is actually a battlefield? A really complex world of microscopic fungal warfare happening, well, right under our noses.
Yeah, it's fascinating stuff.
Today, we're diving deep into that world. We're talking about powdery mildews, specifically focusing on the fungal order Erysiphales, and we're drawing our insights from a fantastic chapter in the classic Introduction to Fungi.
That's right. Our mission in this deep dive is really to pull back the curtain on these pervasive organisms. We want to unpack the intricate biology of these obligate plant pathogens.
Obligate plant pathogens, okay?
Yeah, understand exactly how they wage war on plants, the ingenious, sometimes, well, dramatic ways plants defend themselves, and the surprisingly ancient, yet also cutting-edge strategies we humans have developed to try and control them.
Sounds like quite a story.
Oh, it is. Prepare for some truly mind-bending biological mechanisms and crucial insights into why these fungi are such a persistent challenge,
Okay, let's unpack this then. When we say powdery mildews, what exactly are we defining here? What makes them unique in the vast fungal kingdom?
At their core, powdery mildews are a clearly defined group. There are about 500 species, roughly. And here's the kicker. Every single one of them is an obligately biotrophic pathogen of plants.
Obligately biotrophic, that sounds important. It's a bit of a mouthful, but it sounds like a really fundamental characteristic. Can you give us the quick translation? What does that actually mean for their survival?
It's absolutely fundamental. It means they must have a living host to survive and grow. Full stop.
Right.
Think of it this way. Unlike many other fungi that can, thrive on dead organic matter or maybe be grown easily in a petri dish with just some sugar solution, these fungi are true parasites. They rely entirely on the living cells of their host plant for all their nutrition.
So you can't just grow them in the lab easily.
Exactly. That's why culturing them artificially is incredibly rare.
Yeah.
I mean, there have been a few breakthroughs, like successfully growing Blumeria graminis on agar, but you still need living barley leaves mixed in. Generally, though, if the plant cell dies, they die.
So their entire existence kind of hinges on keeping their host alive, at least long enough to feed. And that distinctive powdery look they give plants, what's actually causing that?
Yeah, that whitish, dusty film you see on infected plant shoots, that's actually caused by the abundant, really rapid production of their asexual spores. They're called conidia.
Conidia, okay.
It's literally millions of these tiny spores just carpeting the surface. In fact, the term powdery mildew is often used interchangeably for both the disease itself and, the organisms that cause it.
Fascinating. So they're basically just covering the plant with their offspring. What kind of plants are typically on their menu? Are they picky eaters?
They mostly go for angiosperms, particularly dicotyledons, flowering plants with two seed leaves. But there's a very significant exception, Blumeria graminis.
Ah, you mentioned that one.
Yeah, you might know it by its older name, Erysiphe graminis. This particular species uniquely attacks cereals and grasses, which of course makes it a major agricultural concern.
Right. So are these just a cosmetic nuisance then, or do they pose serious threats to our food supply or even natural ecosystems?
Oh, they absolutely can cause significant economic problems. Think of Podosphaera leucotricha, which causes severe apple mildew, or Podosphaera mors-uvae, that's responsible for American gooseberry mildew.
Okay.
And maybe the most famous one, Angiosperms necator, that's the great powdery mildew. It almost decimated the French wine industry back in the 19th century before they found a remedy.
Wow.
But on the flip side, you'll also see many species that cause less destructive infections, like Microsphaera alphatoides on oak trees or Phylactinia guttata on hazel. So yeah, they're widespread, but their impact varies wildly.
Okay, so we've got a clearer picture of what they are and what they look like. Now let's go deeper. How do these fungi actually work? What's their structural game plan? And how do they manage to feed on a living plant without, immediately killing it?
Right. So their fungal body, the mycelium, it's generally made up of these single-celled haploid segments. Each cell has one set of chromosomes. Now, crucial to understand is that this mycelium usually stays confined just to the surface of the leaf.
On the surface, not inside.
Mostly, yeah. They're not really burrowing deep into the plant's internal tissues, with a few exceptions. Instead, the infection begins with these specialized structures called appressoria.
Appressoria.
Yeah. You can imagine them as sort of tiny adhesive pads. They help the fungus stick really firmly to the leaf surface, and then they initiate penetration into the plant's outermost layer, the epidermal cells.
So they don't just like chew their way in. They've got a specific tool for the job, this appressorium. And once they're in that outer layer, how do they tap into the plant's nutrients without fully invading the cell itself? Because you said they need it alive.
That's the really clever part. Once they've successfully breached the epidermal cell wall, they don't just burst into the plant cell's main body, the cytoplasm. Instead, they form another highly specialized structure, the haustorium.
Haustorium. Okay, another key term.
Right. Picture it like a finger-like projection that pushes inwards on the host's plasma membrane, the cell's inner lining. It doesn't actually break through it, though.
Ah, so it kind of makes a pocket.
Precisely. It invaginates the membrane, creating this unique pocket called the extrahaustorial matrix. And this is where the magic of nutrient exchange happens. It creates a huge surface area for contact and allows for really efficient nutrient transfer without the fungus fully invading and killing the host cell right away.
That makes sense.
Now, I should say, only a few species, like that phylactinia on hazel, are known to sometimes penetrate a bit deeper, beyond just the epidermis. But generally, it's all happening at this haustorial interface.
That's a truly clever strategy for a parasite keep the pantry stocked by not destroying the pantry itself. So, okay, how do they reproduce and spread so widely if they're so dependent on a living host? Seems like a vulnerability.
They are incredibly efficient at asexual reproduction. It's really their main game during the growing season. Soon after infection, those conidia, the spores that make the white powder, are produced rapidly and just abundantly from these specialized foot cells right there on the infected leaf surface.
Foot cells.
Yeah, it's just the structure they grow from. And depending on the species, these conidia can form in long chains, kind of like beads on a string, or they might form singly. These conidia are the primary method of spreading infection, allowing for numerous cycles within just one growing season. A single powdery mildew patch can release, well, tens of thousands of spores every day.
Tens of thousands daily. No wonder they spread. Now, here's where it gets really interesting, I think. You mentioned earlier these conidia are unusual compared to most fungal spores. What makes them so peculiar? Because I'd imagine they'd need, like, dew or rain to germinate, right?
That's the unique trick. And it's a huge advantage. Unlike the vast majority of fungal spores out there, Erysiphales conidia are fully hydrated. They're already plump with water.
Already got their water bottle packed.
Pretty much. So they do not require any external water, like a film of dew or rain, for germination. In fact, for some species, free water actually inhibits germination.
Wow, the opposite of what you'd expect.
Exactly. They prefer really high humidity, sure, but no standing water drops. They're highly vacuolated, lots of internal water storage, and they store glycogen as their main energy reserve. This exceptional hydration status lets them germinate and infect under conditions that would just stop most other fungal spores in their tracks. It's a major reason for their widespread success.
That's a total game changer for dispersal. Let's zoom in on the infection process itself. You mentioned Blumeria graminis, the one on cereals, is really well studied. Can you walk us through how it orchestrates its attack step by step?
Okay, yeah, let's trace it. Imagine a single conidium of Blumeria graminis landing on the waxy, sort of water-repellent surface of a grass or cereal leaf. Almost immediately, tiny spines on that conidium release what's called an extracellular matrix. It's like a sticky glue containing proteins and enzymes, including cutinases, to deal with the plant's waxy cuticle. That's for initial attachment.
Sticking on tight.
Right. Then, within about 15 minutes, another batch of this matrix material gets released. This actually requires the fungus to make new proteins on the spot.
Yeah.
Then, within about 2 hours, a primary germ tube emerges. Now, here's a quirk of Blumeria graminis. This first tube doesn't form the main penetration structure itself.
Oh, what does it do then?
It acts more like a sensitive probe. It senses the host surface things like how hydrophobic it is, maybe detects some chemical breakdown products from the cuticle. It's doing reconnaissance.
So it's not just crashing through, it's checking things out first. Very tactical.
Exactly. It's assessing the target. After that initial probing, a separate, usually much longer secondary germ tube forms. And this tube develops the complex, often lobed, appressorium.
Okay, there's the appressorium.
Right. And from the underside of this appressorium, a thin penetration peg emerges. Now, this peg uses a combination of tools, secreted enzymes, like cutinases and cellulases, to chemically soften the plant's cell wall, and a surprising amount of turgor pressure.
Trigger pressure, like water pressure.
Yeah, basically internal water pressure pushing outwards, like inflating a tiny rigid balloon.
Ah.
It uses this physical force, along with the enzymes, to breach the host cell wall. This whole penetration process can happen pretty quickly, maybe within about 12 hours of the conidium first landing.
That's fast.
It is. And once that peg is inside, the very tip of it expands to form the haustorium initial, establishing that critical nutrient supply line we talked about.
And just to be clear, that haustorium is truly its only way of feeding.
Absolutely. For all Erysiphales, the haustorium is the sole means of nutrient uptake. And the exchange itself is fascinating. The plant, just doing its normal thing, breaks down its main transport sugar, sucrose, into glucose. This glucose then passively diffuses into that extrahaustorial matrix, that pocket between the fungus and the plant membrane.
Okay.
And from there, the fungal haustorium takes it up using a clever protein gateway called a proton uniport mechanism. It's a highly efficient, really sophisticated system that lets the fungus siphon off nutrients while, ideally for the fungus, keeping the host plant alive to continue providing that food.
It's like a perfectly engineered biological tap.
Yeah.
Okay, so the fungus is relentlessly trying to get in and slurp up nutrients, but plants aren't just helpless victims, are they? Surely they have defenses against these invaders.
You're absolutely right. Plants have evolved remarkable multi-layered defense systems. It's a constant arms race. Even before the fungus fully penetrates, just the initial contact with the conidium and that primary germ tube can trigger a really rapid reorganization of the cytoplasm within the attacked epidermal cell.
Like an internal alarm goes off.
Pretty much. You'll often see a visible halo forming around the point of contact under a microscope. That's where the plant is actively depositing protective substances, things like phenolic compounds, which are antimicrobial, and hydrolytic enzymes directly into its cell wall right at the attack site. This is a general, non-specific, immediate reaction, a first line of defense.
So the plant detects the intruder and immediately starts reinforcing the wall. What happens if the fungus tries to push through that reinforcement with its penetration peg?
That's when the plant might deploy another defense, forming what's called a papilla. This is a localized, really dense thickening that forms between the plant's cell wall and its plasma membrane, right underneath the penetration attempt.
Like a plug.
It's exactly like a plug. It can physically block the penetration peg and prevent the infection from getting established. And these papillae are quite striking under certain types of microscopy because they glow brightly, they autofluoresce. That's due to the accumulation of those antimicrobial phenolic substances. The plant is literally trying to wall off the invader with chemical and physical barriers.
That sounds pretty effective. Is there like a genetic component to how strong these papillae can be? Can some plants build better plugs than others?
Absolutely, and it leads to one of the most remarkable stories in plant genetics and breeding for resistance. In barley, there's a famous example involving the
Melillo.
Yeah, Nello. It's actually a mutation in a specific gene. When barley plants have this particular mutation, they gain broad spectrum, almost complete resistance against Blumeria graminis. And the reason is, this mutation causes the plant to form incredibly thick, robust, impenetrable papillae.
So the plug is just too strong.
Essentially, yes. It makes it extremely difficult, almost impossible for the fungus to penetrate. And what's truly impressive is that this mullo-based resistance has remained stable and effective for decades in various barley cultivars grown commercially. It's a fantastic example of how a single gene can confer durable resistance.
That's a huge win for plant breeders, finding something like that. But what if the fungus, maybe a particularly aggressive strain, does manage to get past the papilla and establishes that haustorium? Does the plant have a sort of last-ditch, more dramatic response?
In resistant cultivars, yes, definitely. The plant can unleash a more specific and often much more dramatic response called the hypersensitive response, or HR.
Hypersensitive response. Sounds serious.
It is for the cell involved. When a haustorium attempts to form inside an epidermal cell of a resistant plant, that specific cell undergoes what's known as an oxidative burst. It essentially floods itself with reactive oxygen species, like hydrogen peroxide and various destructive enzymes.
It attacks itself.
It does. This causes the infected cell to rapidly die. It's programmed cell death, essentially. And since powdery mildews are obligate biotrophs, remember, they absolutely need living tissue to survive. A dead cell is a dead end for the pathogen.
Ah, so the plant sacrifices one cell to save the rest of the plant.
Precisely. It's a strategic sacrifice. Stop the infection right there before it can spread or even feed properly.
And this rapid, targeted cell death often ties into the gene-for-gene concept, doesn't it? That's a big idea in plant pathology.
Exactly right. It's a foundational concept. It basically describes a molecular recognition system. Think of it like this. The pathogen has certain genes, called virulence genes, which might code for a specific protein, an effector. The host plant, if it's resistant, has a corresponding resistance gene, often coding for a receptor protein, like a lock and key. Very much like a lock and key. If the pathogen's effector protein, the key, is recognized by the plant's receptor, the lock, it triggers the defense cascade, and very often, that cascade leads directly to the hypersensitive response we just discussed. This specific recognition leads to strong, often complete resistance against that particular race of the pathogen.
So, armed with this incredible knowledge of how these fungi work, their attack strategies, and how plants naturally defend themselves, how do we humans fight back, especially in agriculture where these can be so damaging?
Well, we employ a multi-pronged approach, really integrating different strategies. One of the most important and sustainable strategies is breeding for resistance.
Using those resistance genes we just talked about?
Exactly. Selectively developing plant varieties that naturally possess these protective resistance genes. A smart technique within this is called pyramiding. Yeah, it's where breeders try to combine multiple different resistance genes into a single plant variety. The idea is to make it much harder for the pathogen to evolve and overcome all of them at once, build a higher genetic barrier, so to speak. Although the constant evolutionary pressure means the risk of new super races of the pathogen eventually developing still exists.
Building a genetic fortress, essentially, makes sense?
You could say that. We also differentiate between different types of resistance. There's vertical resistance, which typically involves those major genes, the lock and key type, providing complete control, but only over specific pathogen races carrying the matching virulence gene. Then there's horizontal resistance.
Horizontal.
Yeah, this usually relies on multiple minor genes working together. It might only offer partial control, slowing the fungus down rather than stopping it completely. But the advantage is that it's often effective against a much broader spectrum of pathogen races. And there's also an interesting phenomenon called adult plant resistance, where certain resistance genes become much more effective as the plant matures.
So resistance can change over the plant's life.
Interesting. Okay, beyond breeding, what about chemical interventions? Fungicides. I've heard sulfur mentioned in old gardening tips. Is that actually a thing?
It absolutely is, and it has an amazing history. Powdered elemental sulfur is by far the oldest known remedy against powdery mildew. We're talking mentions dating back to Homer around 1000 BC.
Seriously?
Homer. Seriously.
Right.
And it was famously rediscovered in the 19th century. When combined with lime forming what's called the Bordeaux mixture, though sulfur itself was key, it literally saved the French wine industry from Uncinula necator, the grape powdery mildew, when that mildew first invaded Europe from North America. So yes, elemental sulfur is still used today. Quite a testament to its efficacy.
That's incredible longevity for a pesticide.
Yeah.
What about more modern fungicides? How do they work? Modern chemistry has given us a really diverse toolkit of fungicides, crucially with different modes of action, which helps manage resistance. We have systemic fungicides, ones that get absorbed and move within the plant. Examples include things like binomol, which messes with fungal cell division, or the morpholines and triazoles. These inhibit different steps in making ergosterol, a key component of fungal cell membranes. Damage the membrane, you damage the fungus.
Okay.
Newer compounds include the strobilurins. These are really interesting. They inhibit mitochondrial respiration, essentially cutting off the fungus's energy supply. And then there's quinoxaphene, which is quite unique.
How so?
It primarily inhibits only the infection-related events, things like conidium germination and appressorium formation, rather than stopping the fungus's general vegetative growth once it's established. And it's not truly systemic, but it works in the vapor phase above the leaf surface, binding to the waxes, perfectly positioned to stop infection right at the start.
Clever positioning. And what about activating the plant's own defenses, almost like giving the plant a vaccine? Is that possible?
Yes, that's a really exciting area. That's where systemic acquired resistance, or SAR activators, come in. Compounds like benzothioneols, for example, don't directly kill the fungus. Instead, they trigger the plant's own natural broad-spectrum defense mechanisms.
So you're boosting the plant's immune system, in a way.
That's a good analogy. You're priming it to defend itself better against a whole range of potential attackers, including powdery mildew. The beauty of this approach is that because you're enhancing the plant's defenses, rather than directly attacking the fungus with a specific chemical, it's much harder for the pathogen to evolve resistance against these activators.
That makes a lot of sense.
Yeah, so the modern integrated approach against powdery mildews Often involves using cocktails or alternating fungicides with different modes of action, maybe combined with SAR activators and resistant varieties,
Oh.
all to slow down the evolution of resistance in the fungal population.
A combined assault is key. Okay, one more angle. What about using other organisms to fight the mildew? A biological approach.
That's biological control. And yes, there are options there too, though often with limitations. One of the most well-known examples is Ampelomyces quisqualis. This is actually a parasitic fungus that specifically preys on powdery mildews. It grows inside their hyphae and disrupts their development.
Fungus eats fungus.
Exactly. While it sounds promising, its practical effectiveness can sometimes be limited by its environmental requirements, particularly humidity. It often needs higher humidity to thrive than the powdery mildew itself does. However, it is somewhat tolerant to certain chemical fungicides, which opens the door for integrated control strategies using both methods together.
Interesting. Any other biocontrol agents? You mentioned something surprising earlier.
Yes. Well, besides Ampelomyces, other fungi have been found to produce biologically active substances, like certain fatty acid derivatives that can disrupt the powdery mildew's plasma membrane. And believe it or not, this is even part of the reason why applications of things like, yes, cow's milk have shown some effectiveness in controlling powdery mildew in certain situations.
Cow's milk, I knew you'd come back to that. Amazing. So it's the fatty acids.
That seems to be a major factor, yes, disrupting the membranes. However, it's really important to note that biological control, especially for airborne pathogens like powdery mildews, which spread so easily, tends to be most effective and reliable in controlled environments, like greenhouses. Out in the field, it's much harder to manage the conditions to consistently favor the biocontrol agent over the pathogen.
Right, the real world is messy. Okay, we've explored how they infect, how plants fight back, and how we fight back. But let's step back again and look at their bigger picture, their entire life cycle and how they manage to spread so effectively across vast distances. Let's revisit Blumeria graminis.
Right, so thinking about the life cycle and spread epidemiology, really these fungi are incredibly prolific asexually. Remember those conidia? A single small patch, a pustule, of Blumeria graminis on a leaf can release something like 15,000 conidia per day.
15,000 per pustule per day. That's an astronomical number.
It really is. And this leads to incredibly rapid disease spread and multiple infection cycles within just one growing season, especially when conditions are right.
And how do all those spores travel? Is it just wind?
Primarily, yes.
Yeah.
These conidia are perfectly adapted for wind dispersal. They're lightweight and can travel considerable distances. We have documented cases through spore trapping and genetic tracking of spores migrating from, say, northeastern England and Scotland all the way over to Denmark in about 48 hours.
Wow, crossing seas.
Absolutely. They are, you could say, nomadic species, constantly moving across continents. And this constant movement, this gene flow, actually influences their genetic makeup and their ability to rapidly adapt to new conditions or new resistant plant varieties.
So they're constantly evolving as they ride the wind currents. That's quite a thought.
Precisely. Now, in terms of the optimal conditions for infection, remember, they thrive in high humidity. Think 98 to 100% relative humidity is ideal, but critically, no free water on the leaf surface. And moderate temperatures, generally around 15-20 degrees Celsius, are perfect. These conditions are pretty common in major agricultural regions like northwestern Europe, making it prime territory for Blumeria graminis epidemics.
And the economic impact of this constant spread, how bad can it get for crops like barley?
Crop losses can be really significant. Sometimes up to 40% yield loss has been recorded in heavily infected barley, which is one of the most seriously affected cereals. The damage occurs primarily because the infected leaves suffer from drastically reduced photosynthesis. They just can't produce enough sugars for the plant to grow properly and fill the grain.
So it weakens the whole plant.
It does. In fact, heavily infected leaves can even become a sink for carbohydrates. They actively draw sugars away from healthy parts of the plant, further depleting the plant's resources and ultimately impacting grain development and yield.
Okay, we focused heavily on the asexual conidia for spread and infection. Do these fungi have a sexual stage as well? And what role does that play in their survival?
They absolutely do, and it's quite important, especially for surviving unfavorable environmental conditions. Later in the growing season, typically as conditions change, powdery mildews form their sexual fruiting bodies. These are called chasmathecia.
Chasmathecia, got it.
For Blumeria graminis, these appear as small, dark brown, almost spherical bodies. You can often see them nestles within the white mycelium on old cereal leaves toward the end of the season. Unlike many other powdery mildews, Blumeria chasmathecia don't have obvious external appendages sticking out.
And what's inside them?
Inside these Chasmothecia are club-shaped sacs called asci. Each ascus contains the sexual spores. When the Chasmothecium is mature and conditions are right, often after a period of wetting, the asci will forcibly discharge their spores. They use a kind of squirt mechanism as the Chasmothecium breaks open.
So they literally squirt their sexual spores out when the time is right? That's quite a dramatic exit.
Yeah, it's pretty forceful. The primary role of these Chasmothecia is thought to be survival. They act as overwintering structures in temperate climates, surviving the cold, or as oversummering structures in regions with hot, dry summers. They protect the fungus through those adverse conditions, providing a source of primary infection for the next growing season when conditions become favorable again.
So they bridge the gap between seasons.
Exactly. Although, for Blumeria graminis specifically, there's also the concept of the green bridge. Because winter cereals are planted in the autumn and summer cereals in the spring, the fungus can sometimes just continuously cycle between these crops throughout the year, never truly needing a dormant chasmothecial stage in some regions.
Always finding a living host somewhere. Okay, beyond Blumeria, you hinted earlier that other species have truly unique tricks, particularly with those chasmothecial appendages that Blumeria lacks. Tell us about some of those special features.
Yeah, this is where the diversity within the Erysiphales really shines, although our understanding of their relationships is changing. Recent DNA studies have actually significantly reshuffled how powdery mildew genera are classified.
How so?
Well, traditionally, classification relied very heavily on their sexual features, the morphology of the Chasmothecia, and especially the type and shape of their distinct appendages. But we're now finding that asexual features, like the structure of their conidia and the way they're produced, the anamorph stage, are often more reliable indicators of their true evolutionary relationships.
Interesting. So the asexual stage is more informative genetically sometimes.
In many cases, yes. It means that some genera that were defined based on appendage types like Spherotheca or Microsphaera and Uncinula are now often being grouped under broader genera like Podosphaera or Erysiphe, respectively, based on the DNA evidence.
So what we once thought were distinct groups based on those fancy appendages might just be variations within a larger group, genetically speaking.
Exactly. The genetic evidence is giving us a clearer, though sometimes more complex, picture of their family tree. But even though their strict phylogenetic value for defining major groups is now seen as limited, those chasmothecial appendages are still incredibly characteristic and often very useful for quick visual identification in the field or lab.
Like visual calling cards.
Precisely. For instance, Uncinula necator, that great powdery mildew, has these really distinct uncinnate or hooked appendages, like little grappling hooks. While Podosphaera species typically have appendages that are dichotomously branched at the tips, like tiny, repeatedly forked twigs, they're very distinctive.
Okay, but what's truly fascinating here, he said, is that some of these appendages have incredibly specialized functions beyond just identification, right? Like a built-in dispersal mechanism.
Yeah, absolutely do. And the example often cited, because it's just so brilliant, is Phylloxera guttata. That's the powdery mildew commonly found on hazelnut trees, among others. Its chasmothecial dispersal mechanism is a fantastic feat of natural engineering. It's truly remarkable for something so tiny.
Okay, paint us a picture. How does it work?
Right, so the chasmothecia form on the lower surface of the hazel leaf. They have two very distinct types of appendages. There's an equatorial group, sort of around the middle, of radiating bulbous appendages. And then at the top, there's a crown of shorter branched appendages that secrete mucilage, a sticky substance.
Bulbous ones and sticky ones, okay?
Now here's the magic. When the chasmothecium dries out, the thin-walled bases of those bulbous appendages buckle inwards, they collapse slightly. This buckling action acts like a lever. It literally pries or levers the entire chasmothecium free from the leaf surface, launching it into the air.
Whoa, so it's not just passively falling, it actively ejects itself, like a tiny catapult.
Precisely, like a tiny self-detaching catapult.
Yeah.
It gets better. Once it's launched and falling, those bulbous appendages then act like little slights or vanes. They orient the chasmothecium as it plummets downwards, causing it to fall like a miniature shuttlecock.
A shuttlecock, seriously.
Seriously. Perfectly oriented, base first. And why does that matter? Because the sticky mucilage is on that crown of appendages at the base. So as it lands, it's perfectly positioned for that sticky base to adhere firmly onto twigs or maybe newly emerging leaves below, ready for spore discharge in the spring when conditions are right again.
That is absolutely ingenious. Nature's tiny, self-launching, sticky shuttlecocks. Wow.
Isn't it incredible? It's a beautifully intricate and highly effective dispersal strategy, showing just how sophisticated evolution can be, even in these seemingly simple fungi.
So from what looks like just a simple white film on a leaf, we've really uncovered this hidden world. It's full of obligate parasitism, intricate cellular warfare fought at a microscopic level, these incredible dispersal mechanisms like shuttlecocks, and of course centuries of human ingenuity trying to keep up, breeding resistant plants and developing targeted fungicides. It's a hidden battleground right in our backyards, constantly adapting and evolving.
That's the perfect summary, I think. Powdery mildews really are a prime example of biological adaptation in action. They showcase that delicate, dynamic, and often dramatic balance between a pathogen and its host. Their unique biology, from those surprising fully hydrated conidia that defy typical spore behavior to their specialized haustoria tapping into living cells. And the complex, sometimes highly engineered chasmothecia makes them a persistent challenge, for sure, but also a constant source of discovery for scientists.
So what does this all mean for you, listening? Well, the next time you spot that powdery white coating on a leaf in your garden or out in nature, you'll know it's not just some random nuisance. You'll understand it as a living testament to a microscopic battle, a masterclass in parasitism and adaptation, and a rich subject for ongoing scientific exploration. Maybe you'll even have a newfound appreciation for simple elemental sulfur and its role in saving the French wine industry. Thank you so much for joining us on this deep dive. It's been fascinating.
It has. We hope you've gained a new appreciation for the often hidden but incredibly complex lives of fungi and the microscopic battles they wage all around us.
Until next time.